Assalamualaikum warahmatullahi wabarakatuh. My name is Adeline Sufi binti Muhammad Azan with matrix number of 208136. So I'll be explaining about the dynamic light scattering first. So about the dynamic light scattering is used to measure the PDI, which is dispersity index, and also the size of the nanoparticle. So, however, um, for the polydispersity index, will be explained by Mariam. So, I'll be focused on the size. The graph is about measuring the size. So, as you can see for the first graph, which is the my SLN, um, which we know as the capsule of the nanoparticle. So, the SLN is actually in a smaller size than mRNA, and also the combination of the my SLN and mRNA, because as we can see in the graph, the intensity of the my SLN is short um, and have so many peaks. So it's shown that the my SLN is lower in size, so they will move faster. So for the next graph, which is the combination of the my SLN and mRNA, um, the size of the peak and the gap of the dip there is longer than the graph in my SLN. So it showed that the presence of the mRNA um, increase the size, where it will increase, it make the movement of the nanoparticle slower than the capsules. So for the third graph, which is the mRNA, it showed that the intensity is big. So it described as the mRNA in bigger size than the other two. So it will move slower than the capsule and the combination of my SLN and mRNA. So for the sentence that below is the description what I have told for the three graph above. So then for the bottom of the graph showed that the size of the each of three types of the nanoparticle, which is the my SLN, showed that it is in a smaller size than my SLN and mRNA, which is the size is bigger than my SLN, and also the mRNA, which is showed the biggest size. Okay, so that is for the DLS. So next, about the polydispersity index, I pass to Mariam.
Okay, hello, my name is Mariam Sakina binti Muhammad Rizwan. My matrix number is 206207. Okay, I will continue with polydispersity index, PDI. Okay, here we can see as the same as components that Adeline have been mentioned around. Which one? The first one is my SLN, the second one is my SLN with mRNA, and the third one is mRNA. Okay, for my SLN, we predicted the PDI will be the lowest because my SLN, as compared to the size of the particles between those three, it has the smaller size, and they are monodispersed particles, which does not have the combinations of large and smaller molecules. Therefore, they have the lowest value of PDI. The next one, we continue with my SLN with mRNA. Even though there are the combinations of two components, however, because of the MySNN does not give a big impact to the sizes when it become agglomerations or combinations, because it's still a nanoparticles. but they are in the form of polydispersed particles and they have an average value. But for mRNA particles, because it is the biggest particles inside the substance, and they have a highest value of PDI, as well as the explanations below has been mentioned, but I just repeat the explanations. The MySLN will have the lowest PDI because it narrow range of sizes, therefore it belongs to monodispersed particles. And the next one is MySLN with mRNA, which have medium polydispersity index because of the mixed ranges of sizes. And then mRNA, wide range of sizes because it exists as a different length sometimes. So therefore it has a highest PDI and very polydispersed particles. Okay, the last one that we will be talking about together is zeta potentials. I'll start first for this part. We have three, mRNA, MySLN with mRNA and MySLN. The first one is mRNA. We predicted it will be have a value of pH nearly to acidic because mRNA is negatively charged particles. While MySLN with mRNA is not neutral but near to the alkali, and MySLN will be alkali because it's electropositive particles. Okay, how do we decide whether the particles will be either acidic or alkali? Because we depends on the electronegativity of the atom. The more electronegative of an atom, the stronger will be the acidic value. Okay, Adeline?
Um, so so the so the PI point is where the charge is known as the zero. So bow. So we need to avoid the PI point because this zeta potential is where we determine the charge of the nanoparticle. So if there's no charge on the surface, we can't determine the zeta potential as we couldn't identify the pH of each of the nanoparticle. So yang dah terangkan ke pasal elektronegatif? Thank you. That's all.
No, stable form.
Belum.
Positive and negativity. Tak ingat tu. Okay, tak apa. Okay, for nanoparticles materials, if we wanted to classify the components as stables or non-stable, we have to look at the voltage value. If the zeta potential value is positive 30 and above, and negative 30 MV below, so it's considered as stable. And the range between negative 30 to positive 30 will be considered as non-stable because it will be easily agglomerate and disperse. Okay, thank you. That's all from us. Continue with the next group.
Okay, next for question D. So there are four material being tested, which are the MySLN, the vaccine, FMySLN, which is the vaccine that is coated with fluorescent, FMySLN containing mRNA, and finally mRNA. Video aku. Okay. Okay. Okay, I am Ikiwaja binti Harun. My matrix number is 208340. So for this question, there are four material being tested, which are the MySLN, which is the vaccine. Second is FMySLN, the vaccine that is coated with fluorescent. Thirdly is the SLN containing the mRNA, and finally is the mRNA. So for the first part of the question, we provide the expected result for for the Fourier transform infrared spectroscopy. So this analysis on the top part. So this analysis can detect the functional group that vibrate when the infrared radiation pass through the four material. So different material have different absorbing or transmitted at different wavelength, which is reflected on the thickness of the graph. So if the material does absorb all the radiation, the line on the graph will be like plain. There's no thickness on the graph. Okay, so the graph we created is only the expected result and not based on actual data. So based on the graph, you can see for the mRNA, it has different thickness when compared to my SLN and F my SLN because they are completely different material. Mhm. Okay, so next, the graph for my SLN and F my SLN, like mostly is identical, except there's a additional thickness presence on F my SLN graph. Like, this is because, like I mentioned, F my SLN is the similar nanoparticle as my SIN, but with addition of fluorescent coating. So each will have extra or additional functional group. Uh, also, lastly, for FMI SLN containing the mRNA, the one in red, will have the combination thickness between FMI-SLN and mRNA as it can detect the functional group from both of these two material. So therefore the FTIR can confirm the presence of the material from this from FMI-SLN and mRNA based on the functional group. So if you notice, like there are some thickness that is not conserved on in red color for FMSN containing mRNA. This is because the thickness is only probably just some background noise. Yeah. Okay. That's all from me. I'll pass to Shasmira.
Okay, thank you, Iqlil. So before that, my name is Shasmira Ganesan and my matrix number is 207323. So if we refer to the case study, so the one before this was FTIS. So the one that I'm going to explain is Annexin VSA. So before we go, actually at the result lah. So before I explain what is the results, we have to understand what is annexin V assay is. So annexin V assay is basically a apoptosis based assay. This assay is to detect apoptosis of cells. So in our case, we used mySLN, F-mySLN, and F-mySLN mRNA and mRNA. So if you guys can see for mySLN and mRNA, there's no results because they are not stained with the fluorescence. And Annexin DSA, they actually use detection of fluorescent microscopy. So without the uh, fluorescent staining, there would not be any results shown. So we go to F my SLN, which showed a green peak. Okay, the green color means it's a living cell. Since F my SLN is just the vaccine, so there's no apoptosis going on there. Thus, that's why it is in green. So moving on to F my SLN mRNA, it is in red and the peak is high is because since there's like vaccine and also the mRNA, so apoptosis will be occurring there, and thus the results is in red. So that is why we obtained this result based on prediction. So that's all from us, from this group. Thank you.
Tunggu Tia Syasmin. I'm having discussion. I can't share. Okay, wait. Okay dah ke? Hi everyone, my name is Siti Aisyah binti Azmi. My metric number is 206335. So we are the next group, which are the team C. We were assigned to perform the cytotoxicity analysis, which are analysis of MTT assay, SubG1 apoptosis assay, and JC1 assay. So the analysis must include concentrations of the vaccine from 50 mm to 500 mm. So our groups use three range of concentrations, which are 50 millimolar, 100 millimolar, and 500 millimolar. So I will explain for the MTT assay, which is the first graph. So MTT assay is the colorimetric assay to assess cytotoxicity. So yellow color will show the cell death and the alive cells will show purple color and formazan is formed. But here we use graph to show the data. So MTT assay can detect metabolic activity of cell. However, it cannot detect the cell that undergoes apoptosis, since cell that undergoes apoptosis also have metabolic activity. So we refer to the graph. If you can see here for the MTTAC graph, the blue color shows the level for my SLN and the orange for my SLN mRNA and the red color for mRNA only. So the graph determine the cell viability or the healthy cells. So here we can see that the highest level for the cell viability or the alive cell is at the mRNA. This is because mRNA did not kill the cells. Then we have the lowest one is my SLN mRNA. This is because the SLN is toxic, and then it tends to kill the cells, and also it can recognize the target cell because it has mRNA and it can kill the cell. Because of that, it has the lowest level of a live cell. And then we have the my SLN, which is only contain the nanoparticles. So the SLN is toxic, but it does not have the target cells. And then from the three concentration, so at the 500 millimolar concentration, we have the lower levels of for the my SLN and also my SLN mRNA because more cells will be killed. So next I will pass to Danu and Fatia for the next essay.
Okay, I'm Danu Krishna and my matrix number is 206886. So basically, now the next assay is known as the SubG1 cell cycle assay, and basically it's a apoptosis assay lah. Okay, so basically, this assay will detect cells that have undergone apoptosis, and this is basically in the stage of the sub-G1 phase of the cell cycle. So the cell, when it's treated with the nanomaterial, it will basically arrest during this the cell sub-G1 phase. So since we have just to give a general view, like when you observe the graph, so basically what will happen is the DNA will peak at the sub G1 as the DNA is being chopped by the endonucleases, and then this will result in like short fragmented DNA with low intensity and then low fluorophore. So this will be detected in the assay. And so when you observe the three graphs there, and since we have three different concentrations, so basically 50, 100 and apa? 150 ke? 50, 100 and 500 ah. Yeah, what was the three concentrations? I dah lupa, I can't read from there.
Yes, 50, 100 and 500.
500. Okay, so basically what we have determined was 50 is such a low concentration. So when our nanomaterial is treated, it will show effect, but then it's not that high level of toxicity. And then at 100 concentration, it's the most optimum, and it will show the highest amount of like cell apoptosis. And then also for the 500, that is extremely concentrated, so it will definitely show like the most and highest level of toxicity. So basically when you observe the first graph under the my SLN only, so basically, it's just the nanoparticle itself. So when the nanoparticle itself is introduced into a cell, it will definitely show a certain level of toxicity. And so basically, we have showed like a high peak for the 500. The highest is 500 at the sub G1. And then moving on to the next graph, which is the nanoparticle itself with the mRNA, we have showed also the quite similar towards the first graph, where there's a peak in also in the sub G1 phase. And then the last final graph is just mRNA alone. So basically, this mRNA alone is not toxic at all when introduced into the cell, because our cells can break it down, and so basically, we have put a completely green line, and because it doesn't induce apoptosis. Yeah, that's all I can pass on to Tia.
Okay. Okay, hello. My name is Siti Nur Fatia binti Muhammad Razin. Contact number 208117, and I'll be explaining to you all about JC1. So JC1 related to difluorescent. JC1 actually exhibit green fluorescent if it only exists in its monomer. And so in order to know that either the cell is healthy or either the cell is healthy or not, it will depends on mitochondria potential. Is it? Um, yeah, mitochondrial membrane potential. So when JC1 aggregates inside the mitochondria, it shows that the cell is healthy. However, however, if However, if JC1 exhibit on its monomer and it exhibit green fluorescent, it shows that the cell undergoes apoptosis. So just now it's been told that we use three concentration, which is 50, 100 and 500 for three things. My SLN, my SLN mRNA and mRNA itself. So the first one is my SLN, which is the nanoparticle itself. So as you can see, there's a 50, 100 and 500 concentration, and 500 have the highest green fluorescent. As JC1 actually detects the fluorescent dye. So that's why it's count every single of cell ada it exhibit red fluorescent or green fluorescent. So actually the graph shown the cell itself. So 500 show the highest one, the highest cell death, while 50 show the lowest cell death. It show that nanoparticle itself is toxic. However, at the 50 concentration, it's too low for the nanoparticle to take an effect, and 500 too high for the nanoparticles to even affect other cell as well. And 100 is like a medical what do you call what do you say? It's a medium effect. I don't know. Anyway, we move on to MySLN mRNA. It even have a highest reading than nanoparticle itself, because this part is where it can detect the target cell as well, and it can kill the target cell higher than nanoparticle itself. So 50 again, it has a lower concentration, so it may have lower effect than 500, but 500 too high, and it may cause other cell death also. And 100 is good for human to take. And the mRNA itself, as you can see, it doesn't affect anything. It doesn't affect at all to the cell, and most of the cell is alive and not dead. So that's all from us. Thank you.
So, hi everyone, my name is Sopita Anak Perempuan Somdet, and my matrix number is 2070, and I and Sri will be representative from Team F, which our question is, your team has been tasked to perform the cytotoxicity analysis aspect for the COVID-19 vaccine using special cells that undergo apoptosis when exposed to the vaccine. In your team, it asks us to brainstorm the expected data that will be generated from three analyses, which is MTT assay, sub-G1 apoptosis assay, and JC1 assay. So for MTT and also sub-G1, I will cover that part, and JC1 assay will be covered by Sri. So the sample analyzed must include three things, which is MySLN, mRNA, which is the vaccine, and also the last one is the vaccine. And the analysis include the vaccine start from the concentration of 50 millimolar to 500 millimolar. So for my part, the first analysis is MTT assay, which is the MTT assay is assay used to analyze the cell cytotoxicity or the cell viability. So our graph is quite different from Chami part, which is we will use line graph. So there is three concentration, which is the type of concentration material, which is the first one is our vaccine, my mRNA, my SLN mRNA. The second one is the concentration of the my SLN, which is the nanoparticle itself, and the last one is the concentration of the mRNA. The graph is self-viability against the concentration of these three types. So for the first graph, it refers to the concentration of the mRNA, which is shown in the blue line. Actually, the blue line is a straight line, but because of the paper itself, it will call, yeah. So as the concentration of the mRNA increased from 50 to 500 millimolar, it did not change the cell viability because the mRNA itself cannot affect the cell apoptosis or the cell viability. The second one is the concentration of the MySLN, which is the drug without mRNA, which the vaccine or the drug still affect the cell but in a very low rate. And the third one is the concentration of the vaccine itself. It will cause the cell to undergo apoptosis in the high rate. Next, let's move on to the sub-G1 apoptosis assay, which is we have three type of graph based on the material itself, which is my SLN, mRNA, the vaccine, and the last one is mRNA. So we can see that when we looking at the graph C, which is the mRNA itself, it doesn't affect on the cell apoptosis, which is the sub-G1 phases. The got three color, yeah. And the second one is we looking at the MySLN, which is the nanoparticle, which is it cause a small effect on the cell apoptosis, which is it quite higher compared to the mRNA. And the last one is the vaccine, which is my SLN. It cause a big effect on the cell apoptosis, and we should also look at the concentration of vaccine, which is in the green color. The highest concentration will give the highest cell apoptosis, followed by the 250 millimolar, and the last one, the lowest one of the vaccine concentration is 50 millimolar. That's all from us. I will pass my part to Sri.
Okay, so for my part, okay, my name is Sri Tinishwan. My metric number is 208405. Okay, I will explain the JC1 essay. So for mRNA and basically this graph, we will observe the red fluorescence and the green fluorescence. So for the mRNA and SLN is both are control. So for mRNA is negative control and SLN is positive control. So we see for the mRNA graph, so why is the red fluorescence is a lot compared to the green? So in mRNA, the untreated cell is a lot, so the apoptosis is a lot. For the SLN, it is empty, so the untreated cell and the treated cells are equal, so that the red and green fluorescence are same. For the SLN and SLN mRNA, it is nanoparticle, so the protein will be inside the SLN, and when the protein is inside the SLN, the apoptosis is less, so that means the untreated cell is less. So the results was the red fluorescence is less and the treated cells is a lot. So that we get the green fluorescence is high in SLN mRNA. That's all from me. Thank you.
Thank you, Savita. Tu je. Okay.
Thank you,
Bye.
everyone. Bye bye. Thank you.
Bye. Thank
And
you.
now
Thank you. Bye bye.